Our next speaker is Daniel Marbach from Roche Innovation Center here in Basel. And the talk is on community challenge assesses network module identification methods across complex diseases.
Hello, everybody. Hello, good afternoon, thanks. So I'm Daniel. I present today the DREAM Disease Model Identification Challenge. It's work that I did during my postdoc at the University of Lausanne. It now overlapped with my work at Roche, Pharma Early Research and Development here in Basel, where I work now, where we are actually also very much interested in these data challenges. Reproducibility is a big issue. As you know, a large part of published research turns out not to be reproducible, as several studies have shown. This is a problem for academia, but also for industry, and I think more generally for society. And reproducibility is key, of course, when you start to work with large-scale data. We have a data explosion in biomedical research, and we, the computational biologists, now have the privilege to play with these data and try to develop predictive models of disease and develop ultimately novel therapies for patients, for example. But as we know, the progress has been really fast on the data side, but it has been slow to really make a difference for patients. So we can do single-cell sequencing of a tumor, but we may still not be able to cure it mostly. Crowdsourced challenges are one way to kind of accelerate research, to promote reproducible research and collaborative research. So in these data competitions, data challenges, you may know the Netflix challenge or the critical assessment of protein structure prediction is another well-known one. You have a data set, it could be omics data, clinical data, in this example generated from a patient cohort, for example. And now instead of a single group analyzing these data, you crowdsource it, and you now have many teams from all over the world. Anyone interested can develop methods, submit their predictions, and now the key is that the organizers now perform an unbiased and objective evaluation of these predictions to really assess how well these methods perform, to perform really a rigorous blinded assessment. And the DREAM Challenges is maybe the leading initiative running such challenges in biomedicine, in translational medicine, and I've been involved in several of these challenges as an organizer, mostly around network inference. There have been many other challenges in prediction of patient disease progression, response to treatment, and so on, sequence analysis. It's very broad. But I'm really excited about this challenge, which is now finished. I'm presenting now the results because it was the first challenge actually focused on network analysis. So there were several challenges about network inference, but once you've constructed your network, you still have to extract biological knowledge from it, right? And this is, of course, equally important as to construct the network in the first place. And I think this is actually the first competition that really assessed methods to identify modules, which is one way to extract biological information from large-scale networks. It's often a key step, right? The whole thing was, of course, a big community effort involving my colleagues at the University of Lausanne, Roche, IBM, and the best performing team, and of course the whole community, the DREAM community, including all the challenge participants. So community detection or network module identification is really a classic problem in network science. Here you see a social network. It's the Game of Thrones characters network, and you can run module identification, so try to find groups of clusters of nodes that are more densely connected than expected in these networks, and they often correspond to functional units in these networks. Here it's the noble houses of these characters. In biological networks, it is well known that modules often group together genes or protein of a related function that participate in some biological process, for instance. So in this challenge, we wanted to evaluate how well different module identification methods perform to extract biologically meaningful modules from these networks. So we had a benchmark including 6 different types of networks, including protein interaction networks, signaling networks, co-expression networks, a cancer dependency, and a homology-based network. Different size, different structural properties, so making for a nice benchmark resource. And we asked participants to run module identification on these networks. And importantly, we anonymized the networks so that they could not use prior information. They had only the network structure. So it was really quite a fundamental question. Can we extract only from the network topology functional modules without any additional information? In the sub-challenge 1, teams run modularization on each individual network alone. It's a classical module identification. And then there was a second sub-challenge where teams used multi-network or multi-layer or multiplex module identification, which has been introduced in some of the previous talks in this session very nicely, and to see if we can refine the modules when they could integrate information across all of these 6 networks or a subset of the networks. And now the key question is, of course, how we evaluate these modules, and we developed a novel approach for this because, of course, this is a very special challenge because there's no hard gold standard, right? There's no really correct module, but we evaluated the methods empirically using GWAS data, and the nice thing about GWAS data is that it's really independent from these molecular networks. So it's really very different type of data that was not used to construct these networks. And thus, if a module shows enrichment for association to a disease in this GWAS data, it's really independent support that this module might be biologically relevant. So we tested each module for association to close to 200 different traits and diseases using these methods that we had developed a few years ago that allowed to aggregate the SNP p-values across genomic regions, across genes, and also across pathways. And so basically the score then was just how many disease-associated modules does your method find. And so I don't have time to go into much detail. We did a very comprehensive analysis, but some of the key lessons are there are top performing methods from different categories, very different types of methods that performed similarly well, but they did not converge to the same solution. So they provided very complementary, very different modularizations of the networks. That raises the question if we can integrate these predictions to form consensus or ensemble predictions. And indeed, you see that the consensus prediction that we derived from participants is a bit better, but we found that it's not very robust, actually. It's only a small improvement, and it only works when you aggregate all teams, but when you aggregate only a few methods, and this is, of course, not practical for users to apply 20 methods. So when you apply few methods, performance deteriorates because these modularizations are just too different. It doesn't make then sense to merge them. So we actually don't recommend the consensus approach. We could, of course, look at performance across these different types of networks. I don't have time to go into detail here, but an interesting result was also that the multi-network or multi-layer networks failed to really improve performance compared to the single network methods. And that was a very surprising, a very sobering result for the people interested in these types of methods. We think the networks might just not have been sufficiently related in this challenge, and I think further work is needed to understand this result. We, of course, also looked at the biology of these modules and could actually show that these disease-associated modules indeed corresponded to core disease-relevant pathways and including, in many cases, known therapeutic targets, and that they could be used to prioritize novel candidate genes for these different traits and diseases that we consider. These are just 2 example modules, but we have many more examples in our paper that you can look at. Just another analysis that we also did was to look at the overlap of disease-associated modules discovered by all these different teams to find which diseases actually share pathways share overlap at the network level, right? And then you can make a disease network where now there are links if the corresponding modules overlap, indicating that these may share some pathways. So the take-home messages are you should really not rely on a single module identification method, and most papers, they really apply just one method. I think this is a quite important message. You should apply methods from different categories and then not merge the predictions in a consensus. And the multi-network method should be used with caution they probably mostly suited when you really have disease or context-specific networks that share some significant overlap or that are more related. And of course also GWAS data are a bit questioned sometimes. Are they even useful to understand disease? Because if you have well-powered GWAS studies, you find thousands of genes that show association, many of them not being really directly involved in the disease. But we've found here in this challenge that when you look at the level of networks, you can then really identify few core disease modules. For each disease, we usually found just a handful of modules that very specifically implicated kind of these core pathways. So all of this is published. You can find all the data, all the tools is available. You can find it on the challenge website. We have a preprint out. It will be published soon in Nature Methods as well. And I also wanted to point out a companion paper that we also just put the preprint out that makes the top performing methods available as Dockerized containers. And finish with a shootout for our talk at the tech track tomorrow. If you're interested to know how more about the types of methods we use now here at Roche in our daily work in drug discovery. Thank you.
We have time for one question. So for the multi-networks that you use, did you check how many of their edges are overlapping? Is it the low overlap?
So it's similar as we've seen in the talk before. The overlap is very low, so it's borderline significant when you actually test if it's significant, but it's low. Yeah.
So given that, I am not surprised.
Even
I would actually expect the multi-network clustering to fail because it works. It will put things into the same cluster if they are densely interconnected in many of the layers, and that's not the case here. And my group has a paper unrelated to your talk, but why this could be and how to potentially avoid it. But I think it's a huge opportunity for the community. So thank you.
I agree. Yeah. Thank you very much.
And our last, but certainly not the least, talk of the early afternoon session will be followed by coffee break before we then continue.